What if you could take a gene from one species and just plug it into another one? Fundamentally change its genetic code. It really does sound like something straight out of science fiction, but it's a reality that's been in the works for decades. Today, we're diving into the science of transgenic animals and the incredible techniques that let us rewrite the very blueprint of life. So let's start with the basics. What is a transgenic animal? Well, at its core, it's an animal whose genetic blueprint, its genome, has been edited to include a gene from a totally different species. This bit of foreign DNA is called a transgene, and the whole process is known as transgenesis. But, the real trick isn't just getting the gene in there. It's making sure it actually works, and maybe most importantly, that it can be passed down to the next generation. And even though this feels super futuristic, the science itself has been around for a while. The journey really kicked off back in 1974 with the very first transgenic mouse. But the real game changer, that was 1982, when scientists finally figured out how to make that new gene heritable, passed on from parent to child. And then, just a few years later in '89, they completely flipped the script. They created the first knockout mouse, where instead of adding a gene, they figured out how to precisely delete one. Okay, so here's the plan for this explainer. We're going to start with the three main tools in a scientist's toolkit. Then we'll look at the different ways they can mess with genes, how they turn them on and off, and finally, we'll see how all this tech is being used way beyond just the lab mouse. All right, so how do scientists actually do this? Let's open up the toolkit. Now, the technology is always getting better, but most of the work creating transgenic mice boils down to three basic methods, and each one has its own set of strengths and weaknesses. Okay, this first method is pretty clever. It basically hijacks a virus's natural talent for injecting its own DNA into a cell. What scientists do is use a retrovirus as a kind of biological delivery drone. They load it up with the transgene they want, and then just let it do its thing. Infect an early-stage embryo. And the process itself is this whole multi-step journey. It starts with infecting that tiny embryo with the viral vector. Then, that embryo gets implanted into a surrogate mother. Once the babies are born, they get screened to find the ones that actually have the new gene. These pioneers, they're called the founder animals, are then bred to create a stable transgenic line, locking that new trait into their family tree for good. But look, it's not a perfect system. The biggest problem is that these little viral drones can only carry a pretty small genetic payload. Plus, there's always a risk that some unwanted viral DNA might sneak in there too. Now, newer vectors, like the ones that come from lentiviruses, they're a big improvement. They can carry larger genes and are a lot more versatile. The second tool in the kit is, well, it's a lot more direct. It's called DNA microinjection. And you can really think of it like performing incredibly delicate surgery on a single cell. It's kind of a brute force approach, but honestly, it's been a workhorse in this field for decades. So how does this cellular surgery actually work? Well, first, scientists gather a bunch of fertilized eggs. Then comes the really tricky part. They take a microscopic glass needle and physically inject hundreds of copies of the transgene right into the egg's male pronucleus. That's the part that holds the dad's genetic code. After that, these engineered eggs are transferred to a foster mom to hopefully start developing. But this brute force approach has some major drawbacks. For one, it is incredibly inefficient. The success rate is often less than 5%. And even when it does work, you have zero control. The transgene just inserts itself randomly into the genome. It's like trying to add a book to a library by just throwing it at a random shelf. Maybe it lands in an empty spot, or maybe it knocks over a whole section and messes everything up. It can have some really unintended consequences. And that brings us to our third and by far the most precise method. This one uses what are called embryonic stem cells. These are these amazing pluripotent cells from a very early embryo, which means they can turn into any type of cell in the body. They basically act like a master key, letting scientists rewrite the genome with incredible accuracy. Now, the process here is a bit more complicated. Scientists take these ES cells, modify them in a petri dish, and then, this is key, they select only the cells that were changed successfully. Then they inject these perfect modified cells into a new embryo from a totally different mouse. The animal that's born is a chimera. It's a mix of cells from the original embryo and the modified stem cells. From there, you just breed the chimeras to get offspring that are 100% transgenic. The real magic of this method is the precision. Because all the genetic editing happens in a dish before the embryo is even made, scientists can control exactly where that new gene goes. This lets them avoid messing up any essential genes. And crucially, it's the technique that makes a gene knockout possible, to not just add DNA, but to precisely remove or switch off a specific gene. So once you have these incredibly powerful tools, the possibilities just explode. We're moving way beyond just adding a gene into the mix. We're getting into a world of much, much more sophisticated genetic engineering. And this brings us to a really important distinction: the difference between adding a gene and replacing one. Think about it like that bookshelf again. Gene addition, the kind we saw with microinjection, is like just shoving a new book onto a random shelf. But gene replacement, that is way more elegant. It uses a natural cell repair process to find a specific book on the shelf and perfectly swap it out for a new one.
In fact, one of the most powerful things you can do with this technology isn't adding a new function at all, but carefully removing one. By turning a gene off, scientists can finally figure out exactly what its job was in the first place.
And there are two main ways to silence a gene. A gene knockout is like physically cutting the power line to a factory. It's permanent. You break the gene right in the DNA. A gene knockdown, on the other hand, is more like intercepting the factory's orders. The DNA, the factory, is still there and totally fine, but its instructions are being blocked, so no product gets made. The gene is effectively muted, not destroyed. And all of this, it really leads to the central theme of modern transgenesis, which is just one word: control. The goal isn't just to get a gene into an animal anymore. The goal is to have absolute, precise control over where and when that gene is active. So, for example, scientists can control where a gene is expressed. By linking a transgene to a genetic on switch, a promoter, that's only active in, say, mammary tissue, they can create a cow that produces a human pharmaceutical protein, but only in its milk. The rest of the cow's body is completely normal. And they can also control when a gene is expressed. It's really like installing a biological light switch. Using these systems like TET-on and TET-off, a researcher can flip a gene on or off just by adding a totally harmless compound to the animal's drinking water. It gives them this incredible real-time control over the animal's genes. This amazing level of control means the applications go way, way beyond the laboratory mouse. The same ideas are now being used on livestock, fish, and birds to create some major real-world benefits in both medicine and agriculture. This technology is literally creating living biofactories. We're seeing it in sheep that are engineered to produce life-saving human proteins right in their milk. We see it in fish that are designed to grow faster and resist disease, which helps secure our food supply. And we even see it in chickens that can lay eggs with valuable medicines right in the egg whites. It's amazing. So let's wrap up. What are the big takeaways here? Well, we've seen how scientists can insert foreign genes using different tools, from those viral drones to straight-up cellular surgery. We've seen that crucial jump from just random additions to precise replacement and deletion. And I think most importantly, we've seen how modern science gives us this unbelievable control, the ability to turn genes on and off in specific parts of the body at specific times. This technology, it's given us an unprecedented power to edit the book of life. The science has handed us the pen. And that leaves us with this huge, profound question that goes way beyond the lab. Now that we have this power, what story should we write next?